In this second part of my talk, I will explain the steps to perform neurofiber analysis. As useful as volume and cortical thickness analysis was, these are simply no longer enough to characterize the changes that occur due to development, neurodegeneration, or neurodisease onset. We must also look at both the quality and the type of connections or the topology that are made. White matter, as shown in the three-dimensional colored segments in this figure, are myelinated neural fibers that are believed to convey information between disparate parts of the brain. Therefore, the quality and connections of the neural fiber tract relate to the ability of the brain to relay information, whereas the volume of gray and white matter relate to its computational capacity. They are both assessing different aspects of the brain's cognitive ability. The steps to perform this computation are very similar to voxel-based morphometry. First, the neural fiber tracts have to be reconstructed, and these tracts must be registered to a reference template brain, because this is the coordinate system on which individual tract-based comparisons will be done. Next, a general linear model that tries to predict typically the density of fiber tract connections or individual fiber tract connections as a function of various kinds of covariates and factors must be created. And finally, a hypothesis testing, statistical hypothesis testing, should be performed to test the significance of this model. Let us begin by taking a brief review into the principles of how MRI imaging can detect the structure of neurofiber tracts. We will use a special imaging sequence known as diffusion MRI. These red spheres represent individual water molecules present within the neurons of a brain. When they come into contact with a magnetic field, which is indicated by this green light direction here, water molecules will start resonating at a specific frequency. And when they resonate, they generate they in turn generate a magnetic field in this direction shown in the blue arrow, which can be detected by circuitry. So our sensitive MRI electronics are able to detect both any variations in the resonant frequency of the water molecules, as well as the directionality of the generated magnetic field of each individual of groups of water molecules. Diffusion, you can think about diffusion MRI as applying spatial patterns of B field towards the water molecules in the neuron. That is essentially marking the water molecules within individual neurons with a temporary marker. Then by allowing some time to elapse, that is by allowing these marked water molecules to then diffuse in the brain, a second measurement is taken, and these two pairs of measurement allows us to assess which direction the diffusion has happened. We are only able to assess diffusion along the dimension at which a B field gradient is applied. And this is the reason why we have something called a B value and a B vector. So in the MRI system, the B value refers to the degree at which we tip this blue arrow as a function of space in the brain, and the B vector refers to all the possible directions in which we assess the diffusion of the water molecules. Water molecules within neuronal water molecules are permitted to diffuse only along the axons within neuronal fibers. Therefore, by measuring multiple B vector directions of diffusion and finding in which part of the brain this diffusion happens, we can reconstruct the many and varied directions of diffusion of water molecules within the brain, which are a proxy for the actual neural fibers. The first reconstruction algorithms were known as diffusion tensor imaging, where in each voxel only one dominant direction of water diffusion was permitted. And by calculating the diffusion direction in each individual voxel and then creating a reconstruction algorithm that starts to stitch together the directions of stitch together to stitch together the primary diffusion direction, algorithms are able to reconstruct tracts known as streamlined. The disadvantage of diffusion tensor approach is each voxel is only able to detect the direction of one diffusion direction. Modern techniques, QSDR and CSD reconstruction, are able to give a model-free estimate of water diffusion. That is, they are able to generate distribution of water molecules in multiple directions within the voxels. So 2, 3, 4 directions. So as you can see in voxels like this one and this one, the modern techniques allow us to reconstruct crossing fibers as well as the dominant fibers, and leads to greater accuracy. And a further advantage of modern techniques is that the measure of fiber directionality, the measure of diffusion, quantitative anisotropy or apparent fiber diffusion, is directly correlated to fiber density, whereas the traditional functional anisotropy of diffusion tensor imaging does not immediately correlate to fiber density and can be affected by various factors like axonal diameter and myelination. I recommend a GUI-based software known as DSI Studio to perform this analysis, but as usual, if you prefer a more powerful command line software, you are welcome to explore MRTrix software, which can be downloaded from this site. The first step is to call up the DSI Studio window, and the reconstruction process is easy. It's comprised of the step to convert the diffusion MRI raw DICOM signals to a .src file, which contains a representation of all the fiber orientation distributions. A connectometry database must be built, which actually looks at all the fiber orientation directions in every subject and reconstructs the fiber tracts and streamlines in each subject, and finally to perform the statistical analysis. So if you look at this window and you select batch processing and the third button under here, which is the batch conversion of DICOM to .src files, this would achieve step number 1. This will bring you to an individual window which shows you first the brain mask, that is this red highlighted region, which is used to only segment out the soft tissue. You may use this button to scroll through the axial sections of the brain and play with these 3 buttons of threshold, dilation, and erosion to get the best possible mask for the whole brain that you want to use for each individual subject. Or you can actually pre-generate by other computational steps a custom brain mask and load it here. This step must be run, will be executed for every single individual in the system. After you're done inspecting the brain mask, click this panel to proceed to the next step. The next step requires us to configure the reconstruction algorithm. Please select QSDR, which stands for Q-space diffeomorphic reconstruction. This is a reconstruction of multiple fibers using the Q-space mathematical technique, and automatically creates the tract representation in an MNI normalized space, which has been normalized to the Human Connectome project of 1021 subjects. Run the reconstruction to complete the conversion process. The next step is the process of building the connectometry database. Please select Diffusion MRI Analysis tab and go to the second option here under connectometry, and this would basically assemble all the .src files within the directory previously generated and build a database, a single database file. In the third step, we will do the statistical analysis. Prior to interacting with the software, we need to generate a text file with individual covariate and factors listed in columns. So the first row would be the name of the covariate factor. In this case, sex is the factor and age is the covariate. And then subsequently, all the entries must be subject matched to the entries in your directory. Once you have this text file prepared, go ahead selecting this tab, Diffusion MRI Analysis, and clicking the last tab here, which is Group Connectometry Analysis. You will be prompted you can load the previous regression files by selecting this button here. Please select multiple regression, and this software performs multiple comparisons using the false discovery rate approach. You can consider this as being analogous to the p-value for single comparisons, and this is your t-value threshold for the t-value statistics. Multiple regression allows us to perform comparisons within a single cohort, whereas if you wish to perform comparisons between two cohorts, please explore the portrait longitudinal change option via reading the instructions online. Thank you for your attention. I'm happy to complete my talk. I'll be happy to hear from you if you would send me an email on questions or queries, and I wish you a good discussion and a good day ahead.